So what's Recce about? Recce is tackling the global health problem of superbugs, and we're tackling that with a broad spectrum antibiotic capable of killing gram-positive, gram-negative and superbug bacteria. But we'll talk more about that in a moment. For those who don't know, what is a superbug and why does it mean anything to you? Well, I can tell you through a real-life example of, in just our own generation, gone are the days where a doctor gives you a prescription and says, go away and you'll be better. They now give you a prescription with many follow-on repeats, and they do that because antibiotics don't work like they used to. So just in our more recent lives, we're already seeing the superbug problem of antibiotic resistance taking place. So we're tackling that problem head on. So let me tell you about it. Usual disclaimer. So Recce is publicly listed on the ASX. We listed in January 2016. We have a broad spectrum antibiotic that's targeting primarily the problem of sepsis. We are a technology, but our lead compound is focused on sepsis. Why sepsis? Any bacteria in the blood is bad bacteria, and that's where we tackle it. And we've actually tackled it to such an extent that the FDA has recognised the progress that we're making in the area. And through that, the FDA has awarded us a very prestigious legal status that we're quite proud about. This prestigious status, awarded by the FDA instead of the TGA, is because that's where 48% of the world's antibiotic market is, about 3% here in Australia, labels our lead compound for fast-track review status for the life of the regulatory process, as well as 10 years of market exclusivity post-approval. Now that post-approval market exclusivity, that's where you get your return on investment. We have patents, they're very strong, they could be challenged. No one's going to challenge a government-backed market exclusivity. So we also have a manufacturing facility currently producing to volumes suitable for Phase 1 and Phase 2 requirements. And we've recently met with the US FDA at their Washington headquarters in the USA, 13 members of their infectious disease department, and this morning made an announcement regarding that, but I don't get into it in this presentation. Quickly on the capital structure of the company, top corner we have the top 5 shareholders. Dr. Graham Melrose, inventor of this technology, who we discuss more later, institutional investor. I'm in there, number 3, other executive director, number 4, and number 5, State 1 Equities. The executive directors have skin in the game. Now, that's not because we're crafty with issuing stock. That's because we invested in the company at day dot. We're the founders. We personally invested. We've actually invested in every stage at every capital raising, and we continue to stand by the business, and we're very proud for that. We have a share price of about 20 cents, around a 20 million market cap. More recently, we can actually see a nice uptick in our graph there. So there's increased market interest coming back to the company, particularly as we continue to engage with the FDA. Here's a little video on it itself.
There have been no new classes of antibiotics on the market for over 30 years. Recce 327 has the potential to break that mould. Recce 327 is a synthetic polymer designed to target bacteria that cause infectious disease, including ones that are resistant to current antibiotics, the superbugs. Bacterial infections of the blood are deadly and affect an estimated 30 million people worldwide every year. This is where Recce 327 is designed to act. Recce 327 is designed to be administered through intravenous injection, so it can quickly spread through the bloodstream. Recce 327 is designed to adhere to the outer cell wall of the infectious bacteria through hydrophobic interactions. This weakens the cell wall, and internal pressures from within the cell cause the bacteria to burst. The body's own mechanisms can then clear the infection. Normal non-bacterial cells remain intact as they do not contain high internal pressures. Tests have shown Recce 327 is fast-acting, and as a broad-spectrum antibiotic, is effective against gram-positive and gram-negative bacteria, including antibiotic-resistant superbugs. Following regulatory requirements for new antibiotics, safety and efficacy tests will continue to urgently explore Recce 327's ability to address the global threat posed by superbugs. Together, we can make antibiotic resistance history. Visit recce.com.au to find out more.
So what is this problem we're tackling? This problem is huge. The problem is actually the number one most expensive condition treated in US hospitals, and about double that of the second most expensive. Sepsis, which essentially is blood poisoning. It happens when bacteria enters the blood from a little scrape or a surgical procedure, and it spreads very, very quickly. For every hour left untreated, the likelihood of survival decreases by 6%. So how big is this market? 30 million people worldwide every year contract sepsis, a third of which die. Antibiotics are not solving the problem, and there isn't actually an antibiotic therapy designed specifically for sepsis. So we're coming into that space as the first antibiotic designed for sepsis, and to date has very good endorsement for treating it. So as an example of why nobody's tackled the space, everybody is focused on natural antibiotics. Natural antibiotics used to work, and they're typically derived from bacteria or fungi, but they work in a specific relationship with the bacteria that they're seeking to treat. One way we discuss it is, if you can picture a lock and a key, here you have the bacteria, here you have the antibiotic. If that bacteria mutates, that antibiotic no longer fits, it no longer works. And that's because that antibiotic is what's found in nature. It's reliant on natural processes. What we've done is we've started from first principles, using a polymer, which is essentially a monomer, or a polymer which is a monomer of many, many beads. We put them together with properties that we want. We're not reliant on what we find. And we have designed that so that instead of being a specific fit, we actually adhere to the outer membrane of the bacteria, cause the cells to separate, and that high internal pressure in the bacteria bursts. As you saw in the video, the remainers of that bacteria is quickly mopped up, out of sight, out of mind. And that is, in simple terms, why we're working and why the others are not, because they keep becoming resistant and they're not tackling that issue. So that's the difference. Here's an example. Now, we've done dozens of studies particularly. This is one we refer to in mice. Why mice? Because they're the closest model to antibiotic behaviour when compared to people. Here we tested against 10 mice. This is Recce. 10 out of 10 survived. 9 out of 10 survived with the other antibiotic, and if not treated, obviously the survival rate's very poor. What's important about this, 9 out of 10, yeah, that's 100 in every 1,000 not living, big problem in the bigger picture. What's more interesting is we chose oxacillin because oxacillin is the best antibiotic for fixing Staph aureus, MRSA or golden staph. We knew it was that antibiotic for that bacteria. With Recce antibiotics, it doesn't matter what type of bacteria it is. This happened to be a gram positive. It could have been a gram negative. It was actually a superbug, but we still would have killed it, and we would have continued to kill it as we did here with repeated use, and that's incredibly unique to us. I like these slides because it's all well and good to do it in animals, done it many, many times, small and large species, but here you actually see at a molecular level what's happening. And I'll walk away from the microphone for a moment just to show you what we're discussing. Here we have E. coli bacteria. Really, really buoyant, under a lot of pressure, very, very deadly. That's at time 0, and these photos, by the way, are by UWA as contracted out, although it's our technology. This is the compound working. We've introduced the compound, it's adhered to the outer cell wall, and you're seeing the bacteria burst. We've got a nice rupture there, you've got another good one down here, and you can see them. That's the antibiotic working. That's causing the bacteria to burst. And you'll actually see after 180 minutes, it could have happened earlier, we just did it in time increments, you've got a terrible splatter of bacteria, no more problem, no more buoyant, viable bacteria, simply mopped up if it was in the person or animal's body, as we've done many times previously, and problem solved. So that's our unique mechanism of action working at a microscopic level. Now, we've presented thousands of pages of data to the US FDA, and the US FDA awarded us this prestigious legal status of QIDP status. That status is a legal status under the GAIN Act, Generating Antibiotic Initiatives Now Act, and it's actually designed to encourage new companies, or to encourage companies and people to create new antibiotics to address this very urgent global health issue. What does it do for us? It labels our lead compound for fast-track designation for the life of the regulatory process, plus gives us 10 years of market exclusivity, as I mentioned, and that's the element I really like. So we've got, under this GAIN Act, we've got open communication with the FDA. If you look up this morning's announcement, we've got a nice photo there in Washington, DC, as they invited us to discuss our unique technology with a 13-member panel of their infectious disease department. So we've certainly got their attention. Here, the obvious advantages. Clinical advantages. What's most important from a clinical context besides the commercial is when a patient presents to the doctor has to determine very quickly what type of bacteria the patient has. It's guesswork. Diagnostics can't determine it quick enough because every hour left untreated, the likelihood of survival decreases by 6%. With this, instead of whacking in 3 or 4 different antibiotics and hoping for the best, in a clinical context, the doctor could prescribe this knowing it will kill a gram-positive bacteria, it will kill a gram-negative bacteria, and it will kill a superbug. So you've got, from a clinical context, the capability of potentially nailing that problem up front before it spreads very, very rapidly. Who invented it? Dr. Graham Melrose, Executive Chairman. He invented the technology. He's ex-Director of Johnson & Johnson and Head of Research for about 10 years. He's a polymer chemist by trade, really knows his stuff. There's then lots of other interesting people. The one I particularly like is our new addition, our non-executive director in the USA, Dr. John Prendergast, who I spoke to just a moment ago. He's chairman of a New York Stock Exchange listed healthcare company. He's also lead director of a NASDAQ healthcare company. He really knows his stuff. And he didn't join us because we gave him a nice pay packet at sort of 50k plus a few shares. He joined us because he sees the real advantage of what we're achieving and the global significance of what may be to come. Problem with antibiotic development, if you get a good antibiotic, everybody doesn't want you to use it because it's very precious. With this, and that's through fear of creating further resistance. With this, it doesn't matter how many times you prescribe it. Antibiotic resistance could be a thing of the past because this does not matter how often that bacteria mutates. It's still bacteria and therefore we still kill it. So that means coming across to the business model side of the antibiotic industry, it could be a scenario that the clinicians could immediately prescribe this antibiotic without fear of it ever coming resistant because the unique mechanism of action does not allow for that. So breaking business models, changing industries, this could be just the ticket to do that. Antibiotic development, heck, there's some interest in the space, big mergers and acquisitions. You can read that separately, but be assured they're big numbers. Manufacturing and production, we're currently manufacturing the product. This is actually it, liquid gold. And quite literally, this thing could be the biggest thing since penicillin by size of market. It's an enormous market that we're tackling and a very significant breakthrough. This product, as you can see, is being produced there. That one's in a drip bag. That's being produced in about an hour and a quarter. To date has been used in dozens of studies, but it's getting ready to go into human trials, and we have the capability of producing that in current volumes for phase 1 and phase 2 purposes. So we're all set up. It's just that ticket to go with the FDA that we're waiting on. As a technology, we're currently focused on sepsis, septicemia or blood poisoning, but the beauty of our manufacture process and as a polymer is we can actually manipulate the structure of the compound itself. We change the molecular structure by increasing the size of the monomers and increasing the length of the polymer. What does that do? It gives us other market applications. Gastritis and intestinal, which is H. pylori, and in the intestines. If you increase the size of the polymer, you can keep it in the GI tract. Now that is very valuable because it still has the antibiotic properties, but you're actually hitting it at the source. So these other applications as a technology are quickly coming up behind our lead candidate and has been recognised by the FDA. So, of course, other fallback positions, animals, huge market, still about 13 times smaller than human market, but again large, and many other fallback positions. Patents and trademarks, yeah, we've got granted patents and more coming. As an overview, yes, guesswork could be a thing of the past, and as a technology, we've got many fallback positions. We go for the highest margin, biggest opportunity, which is sepsis, like I mentioned, growing at 13% per annum, so we're very keen to get in there. And as an investment overview, you can really see that we've got a fantastic compound. We're not doing research for research's sake. We're taking this compound through the developmental phase. We've got a very, very strong team supporting the product. We've got the international regulators, FDA, on our side, 48% of the world antibiotic market there. And heck, favourable landscape. We got a bit of news this morning because they're talking about a $1 billion prize for any new antibiotic developed. It's not awarded yet, but be assured if we've got a string of new antibiotics coming through, it's very motivating to pursue that $1 billion prize. A lot of warm market forces behind us, very capable people in technology, and it's about commercialising it, and that's what we are on a mission to do. Thank you.